talking about gel electrophoresis. Gel electrophoresis is used to separate molecules based on how big they are, usually separating DNA. In our case, we are separating our PET plasmid DNA. If we look into our DNA, we can see that we have a nucleotide. A nucleotide is a building block of the DNA. Connected to the nucleotide, we see that we have a phosphate. This phosphate is a negative charge. This phosphate also gives the DNA an entire negative charge. Now, if we were to look at the gel machine or the electrophoresis machine, we see that there is an electrical field or an electrical charge running through it. Now, this gel is typically made of agarose. In our case, we are using 1% agarose. Now, agarose is known as a polysaccharide polymer, also known as a carbohydrate. This agarose will go ahead and let the DNA molecules travel through it. We see down here that in the agarose, we have one section where there are wells or holes. Within these wells, we will be inserting our DNA or our ladder. This is the area where the wells that are negatively charged, while on the opposite side, the wells or the anodes are positively charged. So, because our DNA is negative, the DNA will go ahead and travel from the negative wells or the negative anodes to the positive poles, positive anodes. In our lab, we are comparing our plasmid DNA that has been cut and inserted with our desired EGFP gene. Because we have cut our DNA into small pieces, we have made smaller pieces of DNA, and when we are inserting them into our wells, we begin to load our samples. If we turn on the machine, the gel will run, and the DNA will begin to migrate from the negative to the positive ends. But because we have cut our DNA, we will see that there will be some fragments closer to the wells, while some other fragments or other bands have led towards the farther side or the opposite side of the machine. This is because larger DNAs or larger DNA fragments carry more molecular weight and tend to move slower, so they would be in the higher end of the wells, while the shorter DNA fragments carry less molecular weight, so they will move faster and appear towards the opposite side of the machine. So we would just begin to see our spread of all of our fragments that have spread through the gel. Now, you can see that these bands are identified for us under a UV light. In our case, we are comparing our cut plasmid with our insert. In lane 2, we have our cut PET with our EGFP insert. In lane 3, we only have our EGFP insert. In lane 4, we have only our uncut, empty PET. And in lane 5, we have our cut, empty PET. We expect to see 2 bands in lane 2 because we have both the DNA and the insert. We expect to see 1 band in lane 3 containing only the insert. Expect to see 1 band in the empty uncut, expecting only the DNA, and the same with lane 5, only 1 band with our DNA. We do see here that we have a DNA ladder. Now, this DNA ladder is useful in order to help us identify our DNA base pair length. We expect to see a band representing our insert EGFP in the 1 kb area, and we expect a cut PET at or in between our 4 and 3 kb area. We distinguish what differences from another using our DNA ladder. Now, our DNA ladder is a sample that contains known fragment sizes. So when we run the gel electrophoresis with it, we already know the fragment lengths. For example, our expectation is to be at least 2 bands in length 2, as stated before, and 1 band in the remaining. Those base pairs are 10, 5, and 1 kb. We would begin with the highest kb pair, knowing that that is the larger fragment, and we will start from the highest to the smallest. But we might ask as to why gel electrophoresis is being used in biotechnology. Well, gel electrophoresis is a step in determining how closely related some species are. So, it can help scientists help distinguish the relation with other species, or it can also help distinguish how closely related you are with your mother and your father genes. Another way that gel electrophoresis helps is through DNA fingerprinting, and this is by identifying or obtaining a DNA sample from a crime scene and using it to compare the likelihood of the victim or the suspect. And this is how gel electrophoresis